In your first cell, how many of you have something like that? That's good, because that's what I see from here. Metaphase, what should they look like? Hey, what'd you just do with yours? Moved them closer together so they were like paired?
Yeah.
In like a specific way?
Yeah, they're too far.
Like maybe like that?
Yeah.
Do chromosomes aligned in a metaphase plate next to each other like that, do they have a special name? They're what? They're homologous pairs. But then anaphase 1 happens. Are they still homologous pairs?
No.
What should I have? Two separate lines, two separate X's. What do you think? How about that? And then I have to move to the next stage, right? The next page. Back on the first slide, if I take a look at prophase 1, take a look at your green X in prophase 1. Are the letters on your green X's the same? So my green X, I'm just going to say it was a capital A and a capital A. And my yellow X, on your yellow X, are they the same?
No.
Are they the same as each other,
Yeah.
but not necessarily the green X?
No. Yes.
Yeah.
So some of you will have one that's the same as the green X, and some of you will have something different, and that's totally okay, so long as each arm of the chromosome has the same thing for the same colored chromosome. So we're going to make mine lowercase a, lowercase a. On your tabletop, it's showing you what? Don't worry, I anticipated that, friends. What should I draw for metaphase 2 of meiosis 2? What do they look like? One X there, and one X here. Then in anaphase 2, what happens to the centromere? It dissolves. So what's in my first 2 set of immature gametes? How many of you made it that far? You're like, yeah, hello, this was meiosis, right? There's something weird about this, though, compared to when you did it. I'm going to fill out the girl ones, by the way, because I'm a girl. There's something weird about this, though, compared to when you did it last time we did this. What's weird?
The colors.
Colors are weird. They're not blue and pink. Should I criticize my husband for his choice of colors? All right. Some of you are like, no. I don't know.
Do they not have like the broken off arm?
We didn't show synapsis or crossing over. We could have, but we didn't because I didn't want you to tear your pipe cleaners.
Do they have different traits on each of the little arms?
So how do you know?
Because of the letters.
So there's a letter written on them. Is that weird compared to last time we did it? Let's go back to the first 4 gametes that you made. Oops, that's a yellow line. Pardon? Undo yellow line. Or white line, sorry. So, the 4 gametes that you made, find them at the bottom of the page. Take a look at the letters that are on them. Those are the 4 potential sperm or eggs, depending which one you made. that your individual would have made. The same colors should be the same letters, right? So, for example, on mine, I should have had, based on what I wrote on my previous slide, my 4 gametes that I could make would be pink pea, or aw, and aw. Do you see how I would have made those 4 gametes? You know where they came from through meiosis? You can follow it all the way through and figure out how you get different gametes. Now that's interesting enough, but a gamete is never going to express a trait. What do I mean a gamete is never going to express a trait? How do you express a gene? If that's true, if what you are telling me, good sir, is the truth, I have 1 chromosome that was labeled capital A, capital A. And I had another chromosome that was labeled lowercase a, lowercase a. And if that label was located in the same spot, do you agree? On both of your chromosomes, they're all located in exactly, actually in all of your chromosomes, they're located in exactly the same spot. In real life, that's the truth. If there is a section of DNA that codes for a protein, hey, what's a section of DNA that codes for a protein? A gene, right? A section of DNA that codes for proteins, a gene. So if there's a gene that occurs right here on this chromosome, it's going to occur in the exact same location in the homologous chromosome. On the sister chromatid, so you agree I have an uppercase A in that location. And I have an uppercase A in this location. Why are they the same? Like I asked all of you, I said your sister chromatids attached by centromeres have the same letter on them, don't they? Every single one of you said yes. Why? Yeah, this is the result of replication, right? This one only exists because this one underwent replication and an identical copy was made. So they should be identical. And in this one, this should be identical to this one because it's the process, it's the result of replication. So on a chromosome, genes are always located in exactly the same spot. And on sister chromatids, they're going to have the exact same copy of each gene. You with me? You see where they are? You're like, I'm okay with this? Let's talk about this copy of a gene thing. Because wait a minute. So A means it's the same gene, right? If it's all A, it's all the same gene. Why is one uppercase and one lowercase then?
Can they express themselves in different ways?
How do they express themselves in different ways? Aren't they the same gene? One could be dominant, one could be recessive. What does that mean?
That one will naturally be shown and one will be not shown.
Okay. I'm good with that.
That when both are expressed, the dominant one is the one that is going to express itself.
So the dominant one dominates the recessive one. Think about this. You get 2 copies of every gene that you have, right? In one copy, you got from mom. In one copy, you got from dad. Did mom and dad have to give you the same version of each gene? So that means that different versions must exist, right? Those different versions of a gene have a funny name. They're called alleles. A-L-L-E-L-E-S. An allele is just a different version of the same gene. In this instance, the two potential alleles are capital A or lowercase a. Now, this dealing in hypotheticals, this capital lowercase thing, seems quite silly. So instead, I'm going to pick some human traits that we could talk about. Human traits that are controlled by different genes. One, hitchhiker's thumb. So you guys, if you're familiar with the fact that when you put your thumb out, if it bends backwards, that's the hitchhiker's thumb. Free or attached earlobes. Whether your earlobe has like this dangly thing, or it just kind of like merges with the side of your head. Free versus attached earlobes. Your ability to roll your tongue or not. So if you can make that little tube-shaped roll out of your tongue, or not, that's another gene. Widow's peak, your hairline. How your hairline comes down on your forehead or not. That's another gene. Now see, all of this, these are boring. I don't like these genes. I would prefer to talk about a gene that I think is funny. None of those made me laugh. It's true. They're all kind of like, meh. There are some very peculiar genes that have to do with how we digest our food. Whether or not you can eat certain foods and digest them or not. So, for example, some of you are familiar with lactose intolerance, right? People who are lactose intolerant got 2 copies of the gene for lactase, the enzyme that digests lactose, but they got 2 copies that don't work. So when they eat lactose, they get sick, right? They're uncomfortable. Gas, bloating, all of the joy that goes with it. That doesn't sound like a fun one to talk about. Nobody wants to talk about people being sick. We certainly don't want to like laugh at that because it would be sad, right? I mean, ice cream, cheese, milk, all good things. Beets. You guys familiar with beets? They're that lovely root vegetable. The Italian version is typically that brilliant red ringed with white. Did you know that they use beets, especially in a lot of organic cooking, because of their brilliant color, that nice red color? You could dye Easter eggs with it. So you can boil a beet, and all of that dye leaches out into the water, and then you can drop your boiled eggs in there and use it as food dye instead of using fake food dye, because you've seen that whole Kraft kerfuffle, right, about the macaroni and cheese causing all kinds of behavior problems in children. Have you seen that? Yeah, yellow dye 5 and yellow dye 6, my friends. Be suspicious. They cause behavior problems. You can find it in Kraft macaroni and cheese. Kraft has pledged to take it out of their macaroni and cheese recipe. You can find it in Cheetos. Really anything with a lovely yellow color, like yellow Gatorade.
Goldfish.
Wait, so is this like an actual proven, or not proven, validated study?
There are quite a few studies that show correlation, but not necessarily causation. But really, who needs to eat food that unnatural yellow color, right? Like, that's not a normal color for food to occur. You want yellow food, it's called a banana. Right? So there are some of us that don't like to allow children to use food dye. So we used beets at Easter at my house to color food, or to color eggs with my nieces and nephews. Because you also don't have to get it hot. You can just drop the egg into the beet juice and there's no heat involved, so then I know that the little ones aren't going to burn their fingers or ingest something toxic or whatever. Stuff you worry about when you're old. So beets, that brilliant pink color. The thing is you need an enzyme to help you digest that brilliant pink color. And that brilliant pink color is water soluble. So if you take a beet and you drop it in a bucket of water, the water is going to turn beet colored because it's a water soluble dye. If your body makes the enzyme that can break down that color, you can eat a beet, your body breaks it down, not a big deal. If you got the gene that can't digest the enzyme, in case you haven't figured this out yet, your body is a what goes in must comes out, like must come out. Like if you eat it, it's got to come out somehow. And there are three exits. One of them we prefer not to use because it ends up being the entrance. Nobody likes that as an exit. Your other two options for exits are for solid waste or liquid waste. But since beet dye is water soluble, it comes out the liquid waste direction. So those of you who got the version of the gene that cannot break down beet coloring, after you eat beets, you pee a brilliant color of pink. Your pee turns pink.
That's a plus.
Totally, right?
That is a plus.
It is definitely a plus. Beets are also very good. They're tasty, they're sweet, that is true. I will actually say that's true. Canned beets are not delightful. Don't do that. Yeah, not a fan of pickled beets. I will roast beets and I will boil beets on rare occasion. I prefer them roasted.
How many people have the ability to drink them?
You want to know what the best part is? Peeing pink after eating beets is dominant. So if you got the inability to digest it, you got the dominant one. So it turns out that, well, even if you got a copy of the I can't or I can digest the beet, you're still going to pee pink. How many of you are like, going to go home and eat some beets? Just so you can find out. So one time I did this with students and I was convincing them that I was going to go home and eat beets just to show them what it looked like. Like, oh, just trust me. Just that way you guys, because they're like, really? I don't believe you. And I was like, no, really, I'll go home. I'll try it. You'll believe me. And I knew one of them worked at the farm market, you know, the Dearborn Farm Market on Monroe. So I purposely went and bought beets and went through her lane. So she was cashier. And then the next day I just brought in some red food dye and I dropped it into some water and then I brought the water in with the red food dye and I was like, see? And she's like, oh my God, she did buy beets yesterday at the Dearborn Farm Market. And then everybody thought I brought in pee and that was hilarious. Well, okay. I thought it was hilarious. They were like, just go home and try it, kids. Go home and try it. Let's talk about that. For the rest of the hour, then, when we talk about capital A, that means I can't digest beet and I pee pink. And when we talk about lowercase a, it means, oh, I don't get to pee pink. So, following your diagram through all the way to the end, you should have made 4 gametes. What does it mean to express? Expressing a gene means making the protein, right? The only way you see what gene you have is if you make the protein. And you don't make any protein if you're not a somatic cell. So, you know, your gametes, they just possess them. They don't do anything with them. They don't do anything with them until what?
Fertilization.
Until fertilization. Can your gametes fertilize themselves? So we are not asexual individuals? No. Okay. We agree on my previous slide. I had 4 gametes, capital A, lowercase a, right? But the 2 capital A's were identical. So I only really made 2 gametes. My choices are, I made a capital A gamete or a lowercase a gamete. Agreed? What gametes did yours make? How many of you have all uppercase from yours? There are some of you out there. When I twisted them together, I made sure some of you existed. So I got one all uppercase. Do I have anybody who's all lowercase? Got a couple all lowercase. And everybody else then, I'm assuming you are one uppercase and one lowercase. Okay. This is going to be interesting. So I've drawn a square on this diagram, or on this slide, and some of you drew this square for me yesterday and you filled it in all nice and neat when I asked you to tell me about genetics. Some of you even labeled it with its name. It's called a Punnett square. And we use Punnett squares to predict the probability of the genes that an offspring is going to have. So for my individual, I would write, well, one of the gametes I made was a capital A. So it goes down the side. And one of the gametes my individual made was a lowercase a. Go ahead and do that now. You can draw a Punnett square. You can do it on your summary sheet on the back and the bottom. There's room for you. And you can draw that Punnett square right on there. And on down the side of it, you're going to write your gametes, the two gametes that you made. So for some of you, that means the two things you're going to write are two lowercase a's. And for some of you, the two things you're going to write are two uppercase a's. But for everybody else, yours is probably going to look like mine. Now, you have to pay attention to the person you share a table with. Because you're going to look at what gametes they made. Ask them. Be like, what gametes did you make? Now you're going to go ahead and write their gametes across the top of the table. So if I had a partner, say my partner was the same as I was, they made an uppercase and a lowercase. Some important things to note about a Punnett square. You're always going to write the uppercase first. So we always write the uppercase first. Now to predict the probability of what genes the offspring will get, we simply follow the gamete to the box. So if my gamete of capital A were to be fertilized by my lab partner's gamete of capital A, the offspring that would result, the zygote, right? That's what happens when fertilization occurs, a zygote. The zygote that would occur would be a capital A, capital A. When we have two of the same allele, so we have two capitals or two lowercases, we call that homozygous. So the zygote would have the same, homozygous. Here, it's homozygous dominant or homozygous capital. What about the individual below that? What would they be?
Uppercase and lowercase.
Uppercase, lowercase, and we call them a heterozygote or heterozygous. And the individual to the left would be another homozygote, right? I'm sorry, another heterozygote. How about the last individual in the corner? What would they be? Homozygous recessive, or homozygous lowercase a, lowercase a. So we have another homozygote. This does not tell me the order that the offspring would be born in. This does not say if these individuals had offspring, these are the guaranteed gene combinations they'd get. This square is simply a matter of probability. It allows us to predict what is most likely to happen. So it's a probability thing. But what we've looked at here is what genes they have. So 1 of 4 individuals is a homozygote dominant. 2 of 4, hey, fractions are our friends. What do I do with that? 1/2 are heterozygous, and 1/4 are homozygous recessive. When we look at the genes that somebody has, we call that their genotype. So my genotype would have been capital A, lowercase a. My lab partner's genotype would have been capital A, lowercase a. And the genotypes of any potential offspring from that combination would be 1/4 of them would be homozygous dominant, 1/2 would be heterozygous, and 1/4 would be homozygous recessive.
The genotype is all the probabilities, right?
Well, the genotype technically is the individual combination of two alleles. So here the genotype for me would be A, big A, little a. That's my genotype.
Oh, the highest one.
Did all of you end up with this as your answer?
No,
Why not?
because I had 2 lowercase a,
And what did your partner have?
uppercase, lowercase.
Oops. And there should be another group who had a different result. Should be that group, right? Who else had a different result? What'd you guys get?
I vote uppercase and he vote lowercase.
Is it possible in your combination to have an offspring who does not express the trait?
No.
Because if you have the uppercase, remember you guys already told me the presence of the uppercase letter, the dominant letter, masks the presence of the other. Now, I'm going to explain this to you guys how this works because it's often misunderstood, and I'm going to use a cookie example. Chocolate chip cookies. We agree there are many recipes for chocolate chip cookies, right? So there's lots of alleles for chocolate chip cookies. And one of them is just a plain old school Toll House chocolate chip cookie. Life is good. It's a plain cookie. But one of them gets all fancy, and it adds peppermint oil, so you get a mint chocolate chip cookie. Have you guys ever used peppermint oil before?
No.
Those of you who are familiar with it, you use very little, right? Why?
It's really strong.
Super strong. So if I made regular old school Toll House chocolate chip cookies, at the same time I made mint chocolate chip cookies, and I stored them in the same container, would I ever know I made the old school chocolate chip cookies? Everything in that container is going to taste like peppermint, isn't it? No matter what you do, you can't avoid it. It's why you don't put your Thin Mints with anything other than Thin Mints. Like they come sealed in that plastic tube. Leave them sealed that way or everything's going to taste like Thin Mint. Same thing here. Mint is good in moderation. Mint's good in moderation. But we understand that mint didn't prevent the other one from being made. Mint simply masked its presence. The other one got made, we just didn't see it or taste it or know it was there. That's how dominant and recessive genes work. The dominant one just masks the presence of the other one. The protein product gets made, we just don't know it's there. It just isn't quite strong enough to be present. So what I want you guys to do before you put those back in your bags is take those summary sheets you picked up, and I want you to write down your gene combinations on that summary sheet, labeling each chromatid with the capital A or lowercase a where it goes. So when we study, we understand how meiosis affects what alleles we get.